Welcome to another whiteboard talk, and in this talk we're going to focus on mitosis, which is a type of cell division, right? And we will be covering the slides in our mitosis presentation. I think there are 3 or 4 slides we'll be covering, okay? So most of the time a cell isn't actually actively dividing. Most of the time it's doing other things. And when we think of a cell's life, then, we tend to draw it as a cycle. We say, all right, a cell has a cell cycle. And this cell cycle has a sliver of that cycle is mitosis and cytokinesis. But the rest of, so we call that the big M phase, if you will. And the rest of the cell's life is called interphase. Interphase. Something like over 90% of its life is spent in interphase. And we can divide interphase into several components as well. We can talk about gap 1, synthesis, and gap 2 as parts of interphase. And now after a cell has just divided, so we had one cell here that gave rise to 2 identical cells, right? Maybe this cell starts out a little bigger. Now these cells go through interphase. They move through interphase, all right? And a few things happen. During gap 1, and I'm going to draw a cell that's going through interphase. This cell is in interphase, let's say, okay? And this cell has a nucleus with its DNA, right? Strings of DNA. I think we're going to have a cell that has 4 chromosomes in it, all right? So that makes it simple. We're going to follow this particular cell. Now, during gap 1, a cell is mostly spending time growing. It needs to grow. It's tiny after mitosis, and so its organelles will replicate, its size will grow, and it will kind of prepare for cell division if it's going into cell division. One other thing that happens during this phase is the centrosome replicates. The centrosome is a section or found in the cytosol of a cell, and it has 2 kind of interesting centrioles made up of microtubules inside of the cell if it's an animal cell. And the centrosome is the microtubule organizing center of the cell. It does the job of organizing the cytoskeleton, those microtubules in the cytoskeleton. Now, if a cell is going to go through, if a cell is going to prepare for another round of mitosis, it's going to replicate its centrosome in gap 1. So not only is it growing, but it also will replicate its centrosome, okay? If the cell is, sometimes the cell leaves its whole cell cycle and goes into what is called gap 0. We'll talk about that again, but a cell in gap 0 is not preparing to go through cell division. It might be just doing its job as a liver cell or a skin cell or whatever it is. It might at some point re-enter the cycle. In any case, after gap 1, the cell is now grown a bit. It's now getting big enough so that it can think about replication. It now moves into synthesis. During synthesis, that means replication. The DNA is replicated, all right? The DNA in the cell is replicated. I'm just going to add in, all of the DNA is an identical copy of it is made. So now we have a cell with kind of 2 copies of all of its DNA. I forgot to add in a few nucleoli, those dark staining regions in the nucleus. But the big part of S is the replication of all of its DNA. We have a cell now that has 4 chromosomes in it. And we'll talk about what chromosomes are in a second, okay? After the cell has replicated its DNA, it now moves into gap 2. Gap 2 is a period in which the cell is still growing, the cell is still preparing for mitosis, but it's also checking to make sure that it's ready for mitosis. There's kind of a safety check. hey, have we replicated all of our DNA properly before we start going through mitosis? Or do we need to stop and, fix up mutations, fix up problems that we had before we get going? Okay? And so gap 2 you can think of as still a growth phase, but also kind of a safety check spot before mitosis can happen. Okay? Now we've got a cell that has four strings of chromatin, if you will, and it has replicated its DNA, it's replicated its centrosome, and now it's ready to go through mitosis. So I'm going to erase my cell cycle drawing, and I'm going to start drawing what happens to this cell in the phases of mitosis. So where is my eraser? I've already lost my black marker. It's somewhere in my house. I'd blame my dog, but he's too old to bother with that. He used to do naughty things, but not so much anymore. Now I'm going to follow this cell as it goes through mitosis. And like I said, I decided to pick a simple cell with 4 strands of chromatin. If we picked a cell like a human cell, it would have 46 strands of chromatin, and I just don't want to go there, right? That's too much. So we're going to think about this cell with 4 strands of chromatin, long, stringy pieces of DNA, right? At this point, that's what it looks like. Now the cell goes through mitosis, and it turns out mitosis is complicated, and we divide it into 5 phases in our textbook. Prophase, prometaphase, metaphase, anaphase, and telophase. So 5 phases, P-MAT, prophase, prometaphase, PPMAT. Sometimes when I'm trying to memorize the phases of mitosis, I come up with some sort of fun mnemonic, like please pass me a tulip, or please pass me a turnip, or something like that. Prophase, prometaphase, metaphase, anaphase, and telophase. And so we're going to draw out what happens in each of these phases. But don't think of them as being static. They're dynamic. The cycle is going to the cell is going to move through all of these phases, and they're dynamic. We've just divided the cycle into, or the process into phases to help us understand what happens first, second, third, etc. Okay, so we've got this cell, and in prophase, and I've got my notes just to make sure that I catch all my notes. We've got a nucleus, right? And we've got the centrosome that's been replicated already. I want to draw that in because that's going to be important. I'm going to write down some things that happen in prophase. The first thing is that this stringy chromatin, the chromatin starts to condense and coil up on itself and shrink up so that it's easier to move around, frankly, for cell division. So the chromatin condenses to form chromosomes. All right, so the chromatin condenses to form chromosomes. And when we look at the genetic material now, we see something that might look like this. Okay, this would represent 1 chromosome. Okay. But it's made up of 2 identical copies of itself because the DNA replicated back in interphase. 1 chromosome made up of 2 identical copies. Each identical copy is called a sister chromatid. Right? So there are 2 sister chromatids, and they're connected. They're connected at a region that we call the centromere. Everything seems to start with C's in this lecture. The centromere is a region that's sometimes near the middle, sometimes it's near the end of the chromosome. It depends on the particular chromosome. And at the centromere is a protein complex called the kinetochore complex. And that protein complex is, each chromatid, each sister chromatid actually has a kinetochore complex. It helps, it's important during the movement of these chromosomes, because really what's going to happen is we're going to move the chromosomes around during cell division. So that kinetochore complex is at the centromere, and there's an enzyme a protein, I should say, called cohesion. I told you everything starts with C. Cohesion protein that holds these two sister chromatids together so that we've got one chromosome made up of two sister chromatids. Now you can think of that, you can make your own setup here pretty easily. I use pipe cleaners. So for one chromosome, one string of genetic material, during interphase it gets replicated. So we have a copy of it. During prophase of mitosis, the genetic material coils up so that we can see them as chromosomes, and these identical copies are stuck to each other at the centromere by this kinetochore complex. All right, so I've got one chromosome made up of two identical copies right next to each other. Eventually, the cell is going to separate these identical copies so they end up in two different cells. Now we have 4 chromosomes in our cell, so I need to draw them in. I will have, in my drawing, I will have 2 long chromosomes that are very similar in size and shape. One I think I'm going to draw red and green. And then I will have 2 short chromosomes, represented here by my 2 short, if I can get them together, 2 short pipe cleaners. Okay, so I'm going to have 4 chromosomes made up of, remember, each chromosome has a replicate, so it's made up of 2 sister chromatids. Let's draw these in, and we're going to follow these chromosomes during mitosis. So I'm going to draw each chromosome. I'm going to kind of cheat a little bit. I'm going to draw each chromosome as 2 lines with a circle between them, 2 sister chromatids with connected at the centromere. Okay, and I happen to have 2 long chromosomes and 2 short chromosomes in this cell. We'll talk about that with meiosis. Okay, so our genetic material has condensed to form chromosomes. That's our first step. Whew, we have a lot going on here, guys. Now, a couple other things that happen. The nucleoli that are found inside the nucleus of the cell start to disappear. Okay? Mine are already gone. I haven't put them in. The microtubules of the cytoskeleton kind of start to fall apart or disassemble, and they are used to start building a mitotic spindle, a spindle of microtubule fibers extending between and from the centrosomes in this cell. So you get this set of fibers extending from each centrosome toward the other centrosome, and that is called a mitotic spindle. The mitotic spindle begins to form, and it's made up of these microtubules that were originally from the cytoskeleton. Okay, that's all I have. Oops, that's all I have written down for the basics of prophase. Now, what happens to this cell? Well, this cell is going to go through prometaphase, so we need to draw in prometaphase next. I have to do some erasing, so I'm going to erase a minute. We're going to follow our cell into prometaphase. This is a phase that you might not have heard about before. You probably haven't. So we've got prometaphase, kind of between prophase and metaphase, right? So let's write down a few things that happen during this phase. And I'm going to take my notes out just to make sure that I don't forget anything about prometaphase. Okay, 2 things basically that are going on. The first is that the nuclear envelope ruptures. So the nuclear envelope ruptures. That means it bursts, right? And so we get a breaking of that nuclear envelope, like that. I need to put in my chromosomes, but I'll do that in a minute. The second thing that happened is the mitotic spindle that's growing enters the nuclear area. So the mitotic spindle enters the nuclear area, because now we don't have a nuclear envelope anymore, right? So I'm going to draw that in Maybe I'm going to draw it in with a different color marker if I can keep track of all my markers. So what happens is these the centrosomes kind of start migrating to opposite ends of the pole of the cell, or poles of the cell, and their spindle fibers are growing and extending inward into the nuclear area. Now, as they extend inward, there are different kinds of fibers, and we need to talk about those two types of fibers. There are kinetochore fibers. Kinetochore fibers, spelling it wrong here. Kinetochore fibers do the job of connecting to the kinetochores of each chromosome, and they connect in a special way. So if I've got a chromosome, and let's just take a green chromosome here. If I've got my green long chromosome with its centromere and its kinetochores, the kinetochore fiber